Welcome. Welcome. Have a seat. We'll get started because it's 2 o'clock. Have a seat, everyone. We'll get started. It's 2 o'clock. This is the mic. Welcome. Have a seat, and we'll get started. It's 2 o'clock, and as you know, in PNB, events start on time. Thank you. It's a thing. It's a tradition. We do that. We start on time. Okay, so before McMaster University was here and before PNB was here, the Haudenosaunee Confederacy and the Anishinaabe Nations lived and traveled and hunted here. In the Haudenosaunee tradition, elders hold respected positions within their communities. The elders are the knowledge keepers, the ones who impart knowledge and tradition and help nurture the next generation. The term elder is bestowed on an individual by their community because of the knowledge that they hold. Knowledge keepers are not self-taught. They've been gifted wisdom and knowledge by other elders and knowledge keepers over years of mentorship and teaching. Part of the Haudenosaunee Thanksgiving address thanks the teachers who've come throughout the ages. Today we're celebrating our own knowledge keepers who've passed not just scientific traditions, but values and ethics and life lessons. And we're benefiting today from the mentorship and the wisdom that they've passed on to the next generation. Whoever you are, wherever you're from, whomever you love, you're welcome here.
So, thanks, Mel, and it's wonderful to see all the people that are here today and so many people I haven't seen for a while. It's really great. And really, it's an honor to introduce this symposium celebrating the amazing achievements of Daphne Maurer and Louis Schmidt. I'm going to be really brief because we want to hear from the speakers. But it's just been so enriching to have these two as colleagues over the decades. So when I joined PNB 32 years ago, Daphne Maurer was my role model. Her endless curiosity and experimental rigor were inspiring and shaped my own career in so many ways, and I will always be eternally grateful. Outside of work, I'll also never forget the musical evenings that she and Charlie used to host in their home. And her Order of Canada is just the culmination of many awards and honors that she has received, and we're so happy to be able to celebrate with her today. You'll hear a lot more about Daphne tonight at the Buttery, so I'm just going to turn now to one of Daphne's greatest legacies, her training of a generation of scientists in the field of visual development. And now we get to hear from three of them. So our first one is Cathy Mondloch. Cathy actually completed her PhD in sibling competition for food in pigeons at Indiana University. I don't know how many of you knew that. But finally she saw the light when she came to Hamilton. She switched fields completely and did a postdoc with Daphne, studying face perception in babies, children, and adults. And it must have been a good experience because after the postdoc, Cathy stayed in Daphne's lab for 12 years as a research associate, together with Terry Lewis, who is here as well, who actually spent most of her career in Daphne's lab. And I will never forget that powerhouse trio. So Cathy eventually went on to a professorship at Brock University in 2004, where she's continued to do really highly recognized research on the perception of emotional face expressions, face recognition, and first impressions. So she's going to tell us about some of that interesting work now. So welcome, Cathy Mondloch.
Haven't taught in person since 2020. Forgot how mics work. So this was a great opportunity for me to reflect back on 12 years that we spent working together and all of the work that we did. And I was told to make it a research-oriented talk and talk about my work with Daphne and my work at Brock, but I couldn't do both because we did so many things together. I'm going to focus on the work that I did here with Daphne and just hint at subsequent research. And as I said to somebody, if you want to hear what I do at Brock, you can ask me to come do a colloquia. But the first thing about Daphne is Daphne thinks outside the box. If I got a letter today from somebody saying, I've studied all of these species since second year undergrad, and now I want to learn about human development and face perception, I'd wonder if they knew who they were even writing to. But that's the kind of letter I wrote to Daphne. I said, I'm looking for a part-time postdoc. I want to move back to Ontario. I resigned from a tenure track position. I want to come to Ontario. I want to learn about human development. Do you have space in your lab? And we agreed to work together for a year so I could get myself started, and we continued to do that for 12 years. Now, we did various projects, but I want to focus on our work today on face perception, which started in 1995. I was just coming off a maternity leave, and Daphne called to say that she got this Human Frontiers grant, and she thought this topic might align better with my interests in development and evolution. And this was a very, very powerful grant because it was a collaboration between Daphne and 3 world-renowned researchers from the US, the UK, and France. And I remember that week, I was literally coming off mat leave. Again, I knew nothing about face perception. I now knew something about human development. And I'm in a room with all of these scholars, didn't know what the terms meant, and I'd go home at night and look things up. But it kicked off my career, and I've been addressing questions about face perception ever since. And so today I want to just tell you about 3 projects that we did together and a little bit about each of them. But these questions were the questions that drove this whole research program. How does face perception develop in the typical population? And then what's the role of experience in driving that development? And when I was thinking about this, I pulled out my last couple of grant applications, and these are still the overarching questions of those applications. So definitely had an influence. So the first of 3 projects I want to talk about had to do with face detection. And actually, I'm really glad Terry's here because she was key to this and did a lot of the testing. I remember you leaving a hairstylist mid-style when a baby was born, so that's dedication. So why face detection? Well, if you think about development, if a baby from birth orients towards faces preferentially, if they like to look at faces, that means that they're playing an active role in their own development because they're giving their developing brain lots and lots of experience with this important social stimulus. So we wanted to first of all know, do babies orient towards faces? And then how does that preference change during the first few weeks of life? So we already knew a bit about this because other labs had done this kind of work, but they had used different images, different ages, different methods. So there was no coherent story. So we collected images from different groups, and then we adapted a method that Terry and Daphne had used to study basic vision, vision acuity, to ask babies what they like to look at. So we would just hold up cards like this. There's a little peephole in the middle, and we'd look through and try to guess where the face was based on the baby's behavior. And so the top card there, most adults say that the one on the left looks more like a face because those 3 blobs are arranged like facial features. And then on the bottom, the face on the left is positive contrast. And we adults find that one more face-like than the photo negative version. So we tested babies right at birth within 2 hours of being born, and then at 6 weeks and at 12 weeks. And what we found was that right from birth, babies prefer to look at that face-like stimulus in the top pair. The youngest baby was 7 minutes old and showed this preference. But the preference for that disappeared by 6 weeks of age. Babies no longer find that to be a very good face. But by 12 weeks, they showed a strong preference for the face on the left, the positive contrast over the negative contrast face. So there's a change in preferences over time. So this was actually our first publication together, so I had to highlight that. And then, of course, what we wanted to know was, well, what's leading to that change? Is it experience with faces, or is it just maturation? So of course, the work that Daphne's probably best known for is her work with cataract patients. And so those are who we turn to. So this is actually my new little grandbaby, looking, of course, at black and white things. And Claire got visual experience right from birth. So her brain is getting experience with faces right from birth. But babies born with a cataract in both eyes don't get that experience. These cataracts block visual input. And so for these babies, there's no patterns out there in the world, and for our purposes, there are no faces. So they're being deprived of that until the cataracts are removed and they're fitted with contact lenses. So for this baby, visual experience starts maybe at 2, 3, or 4 months of life. So Daphne has a wealth of studies examining these patients, looking at what is the effect of that deprivation. And in the study I'm going to tell you about, what we did was test babies on the day that the contact lenses went in. So the first time they get to see, and the question was, are they like normal newborns, which would be an experience is causing those changes, or are they like other babies the same age? It took years to do this study because work that Daphne and others did showed that missing experience is a very bad thing, and so the treatment kept getting moved earlier and earlier. So Daphne almost like scuttled this study because we needed babies to be at least 5 weeks of age at treatment. But over many years, we were able to test 7 patients. They were all more than 5 weeks of age. 4 of them were more than 12 weeks. So are they like normal newborns? Yes, they are. So as a group, these babies preferred those 3 blobs as facial features at an age when typical babies would no longer do that. And not one of them showed a preference for the positive contrast face. So this was really neat because it showed that experience is what's driving this postnatal change. So there are more studies on this topic, but I'm going to leave it there to move on to the next one, because if you think about face perception, we don't just want to say, oh, there's a face. We want to perceive information. We can judge how old somebody is, how they're feeling, and who they are by looking at faces. So this next set of studies, I don't think she's here, but they were done with Sybil Geldart, who was a PhD student in the lab. And so that you can appreciate this, this was done when digital cameras were brand new. And you had to download to a computer, and it took several minutes per picture. And we needed hundreds of pictures. So I think Sybil and I spent months in the basement putting surgical caps on people, getting these images. And what we did here was we developed 5 tasks to measure how children perceive faces, or what information children can perceive. So the tasks were all the same. A face would show up on the screen, that's the target, and then 3 test stimuli would show up, and the kids had to say which one matched the target. And for 2 of the tasks, the job was to match based on identity. So in the top row, you have to find the same person despite a change of point of view, and in the bottom row, you have to find the same person despite a change in head orientation. So this is a very tough task, and actually it's interesting because when I was thinking about this talk, this is the kind of work I'm doing now where I'm asking how do we recognize somebody when their appearance changes. The other 3 tasks, there's all different people being shown, but you have to find the person who's showing the same expression, mouthing the same vowel, or whose eyes are looking in the same direction despite changes in head orientation. So here we tested children, and the children were 6, 8, and 10 years of age. And we also tested adults. And I'm not going to go through all the data, but here's our 2 identity tasks. I just put them together, and then the other 3 tasks. And you're seeing adults there on the far right of each cluster. All right? And what you can see is even adults struggle a little bit with identity, but they're doing quite well. The important point that you need to remember is that the age-related change is much slower on the 2 identity tasks and in the eye gaze task than it is on matching expressions or lip reading. And what I really want you to remember is that the ability to recognize identity is slow to develop. And this is something I'm still trying to understand to this very day. So now that we know that, we thought, let's see how cataract patients do. If something's slow to develop, you might think, well, it doesn't matter if you miss early input, there's lots of time to catch up, right? That would be logical. And so now we're going to go back to these same two groups of babies, but we're going to test them when they're adults. So now both groups have had years and years and years of experience looking at faces, but one group missed experience for just a little bit of time early in development. And it turned out that mattered. So I'm just showing you data from the two identity tasks, and each of these dots shows you the performance of one patient. And any dot below this horizontal dashed line is below the age norms. All right, so they're doing worse than typically developing people. And what you can see is that these patients are doing worse. Right? They're struggling to recognize a face, even though they've been looking at faces for a really long time. Now you might think, well, but wait a minute, those tasks were hard, so maybe patients just don't do well in anything that's hard, but they do normally on the gaze task, which was also hard for children. So this tells us there's something about identity that really depends on that early experience. And again, this has applied implications, right? It pushes the system to get these kids treated earlier. So the next thing that we talked about, and I distinctly remember sitting on a plane, I don't know where we were going, but talking about, well, how do we recognize people? And drawing pictures and all sorts of things. So let's think about recognizing Daphne. All right? We can all recognize Daphne in these pictures. You don't have very many pictures online, but I found these. So we can all recognize Daphne. How are we doing that? All right, what are the cues? And what we figured out was that there were probably 3 cues that people could use to recognize faces. And so we developed what we called the Jane task. And I can explain the name why I've got the Jane task later, but always be careful when naming tasks, because it's now very famous, and we probably could have given it a better name. But Jane is the face on the leftmost face on each of these rows. So that's Jane. And what we did was we created 12 sisters for Jane. And we created the sisters in specific ways. So the top row, we just moved the features around. So move the eyes out, in, up, or down, move the mouth. So one cue we thought of was feature spacing, right? Where are the features on the face? Then we thought another way you might recognize somebody is by the appearance of their features, like the shape of their eyes, the shape of the nose. So we cut out Jane's eyes and we cut out her mouth and put in the eyes and mouths of different women. And then we thought the other thing was the shape of the face. So cut out the inside of Jane's face and we plunked it into different head outlines. And people's job was to just tell us whether they were seeing the same sister twice or 2 different sisters. And again, we tested the same age of kids, 6, 8, 10, and adults. I'm not going to show you data, but just tell you that what's really slow to develop is sensitivity to feature spacing. So 6-year-olds and 8-year-olds were adult-like or nearly adult-like when recognizing faces based on contour and feature shape, but 14-year-olds did worse than adults on feature spacing. So it stands to reason then that maybe feature spacing or sensitivity to feature spacing is important for face recognition, and that's why face recognition is slow to develop during childhood. So, of course, we turn to the cataract patients. And this was work done, I should say, with Rick Legrand, who's now a professor at West. And what we asked was, well, does sensitivity to feature spacing maybe depend on early experience more than sensitivity to features and contour? So we tested, again, patients deprived of vision, completely deprived because they have a cataract in both eyes. They're normal on the contour task, normal on the featural task, but they perform very poorly on the spacing task. So you can see almost all the dots are below that dashed line, which is showing you normal performance. So that's really interesting. Something that takes 14 years to develop depends on early experience. And the other thing I should say is how long you were deprived for didn't matter. 2 months is enough, and you have permanent deficits. So that was exciting, and that got us a publication in Nature. And I remember we were both so chuffed. We went to CNS, and Nancy Canwisher was giving this big talk, And she talked about our study 3 times, we counted. And we're like, whoa, Nancy Panwisher's talking about us. That was awesome. So we thought, though, that that meant that early experience with faces matters. But then Daphne had a brilliant idea. Happened on the coast out west. And I think you were partly like saying, well, there's these other patients that have a cataract in only one eye. And what could we do with them? Because there's a lot of them, and it's got to be interesting. And then she thought about this, and I got to try to explain this to you. But before I show you the picture, the idea here is that if you have a cataract in only one eye, you're seeing faces right from birth, right? I'm just seeing faces like this. But Daphne figured out it might matter which eye you're seeing faces with. Now that might seem strange, except there's 2 peculiarities of the baby's visual system. So consider a patient that has a cataract only in their left eye. That means all their visual input's coming from over here, because they don't see much over their nose. Their noses are small, but they don't see much over there. And that input's going to the left hemisphere. And the other thing that we knew is that there's no transfer of information across the hemispheres during those first few months, which means for these babies, the right hemisphere is deprived. And we knew that the right hemisphere was really, really important for face processing. So Daphne predicted that these babies might do as badly as babies that had a cataract in both eyes, right? Thinking outside the box, she hired me and thought of this. 2 good examples of that. All right, so on the contrast, a baby with the cataract in the right eye, the input's going to the right hemisphere. And so we ran the study, and it turned out, bang on, these babies looked just like that. I don't even have to show you the data. And babies with the cataract in the right eye performed perfectly normally. So this is depth, right? This is taking it to the next level and really trying to get at the mechanisms. We looked at experience in other ways as well. We published research showing that both children and adults are not very sensitive to feature spacing in monkey faces or in faces of a different ethnicity. And so that tells us that the perceptual system is being tuned by our environment and that our expertise is limited to faces that we encounter every day. All right, so I'm going to end. I started with the first study that we did together. This isn't the last study we did together, but it's the last study I did while I was a member of Daphne's lab. And this one has special meaning for lots of reasons. So Gal will understand this. When we say that 6-year-olds aren't very sensitive to feature spacing, they were above chance. And so we decided to ask, well, when does sensitivity to feature spacing first emerge? When do kids first start to be able to use this cue at all? And this was actually an honor student. I love this study, and we have so many storybooks now in my lab because it's a way to get little kids to do a very prolonged study because they like storybooks, and they like magic wands. So what we did was have 2 children come into the lab, and we took many different pictures of them. And then we used them to write a storybook about Johnny and Susie and their day at the farm. And I was rereading this paper thinking about this talk, and I thought, I was so close at this time to what I'm doing now, which is how do faces become familiar and the importance of variability. But I didn't know I was thinking about it when we did this study. And so there are lots of pages in this book. Kids love this book. And what we did was they had them come in the lab to pick up the book. And when they picked up the book, we took a picture of the child. And then we asked the parents to read the book at least 10 times over the next 2 weeks. The idea was we wanted them to become familiar with these faces because we thought if they're ever going to show us they can be sensitive to feature spacing, it's going to be with familiar faces. Then what was neat was when the kids came back, we told them that Johnny and Susie were in a daycare, and the daycare was going to the zoo. And did they want to go to the zoo with them? And they're like, yeah. And then we gave them a magic wand because we learned at a conference that children get bored if they're pointing, but they'll do this all day if you give them a magic wand. And so their job was to wave the magic wand over the real picture of Johnny so that Johnny could go on the train ride. We did this with Johnny. We did it with Susie. We did it with the child's own face, which should be highly familiar. And we manipulated whether it was a spacing, a feature, or a contour change. And what we discovered was that the 4-year-olds make almost no errors when we changed out the contour features, completely at chance for feature spacing. They don't use it at all, not even for their own face. And later I did this at Brock, where I tested kids with their best friends from daycare, perfectly at chance. So something's happening between 4 and 6 years of age, but this is something that develops really slowly and yet depends on early input. Okay, so that's all the studies I'm going to tell you about, but so you know the breadth of Daphne's work. We did the same kind of thing with holistic processing, again with Rick. We studied different aspects of face perception, including a really neat study on perceptions of attractiveness. We looked at different populations, so we looked at individuals with prosopagnosia, and with Lewis Schmidt, we looked at children who were shy. And we wrote many different theoretical papers about development and about face perception. And this one I looked up just for fun, now has over 2,500 citations. So this has really, really impacted the field. And this is where we sat down and just tackled, so what is this thing we call configural processing? So I still study faces to this day. I still have these same overarching questions, but you can fill in the blank and have 3 or 4 more talks about Daphne's work. I could do this about motion perception. Well, I couldn't because I don't know about motion perception, but somebody else in the lab, Terry, could. You could fill in contrast sensitivity, visual acuity. Daphne has had a tremendous influence on our understanding of vision, of face perception, of human development. She's impacted theory.
Thanks, Cathy. Don't take it off. I think we have time for one question, maybe 2.
So when you showed the 2 contrast faces, I wondered, you didn't say anything about race. So it might be that kids that are black or brown might look at that highly contrasted face a little differently. So did you control for that?
We didn't control for that, although I remember we had that conversation, and we were quite convinced, at least at that time, that they would still be perceived as photonegatives. But we didn't control for that. But virtually every baby we've ever tested at 12 weeks, it's the one card, because you also, when we did that study, there were many more cards. You never knew what card you were showing. You always knew when you were showing that card. And every single 12-week-old went to that side. Yeah.
Thank you.
I'll ask a question. So does it matter if the faces are dynamic and moving?
Good question. So we've thought about doing this, and then it keeps getting scuttled. But yeah, it's an interesting question. Do we get form from motion, and would children learn or recognize faces better if they're moving? I've had this Harry Potter, Harry Potter where the faces move in the picture frame in my mind for so long, and I've just never done it. We don't know. And we've done more work on whether it matters if the faces are familiar. So how do kids recognize somebody they know as compared to lab-trained faces? But to my knowledge, there's not a lot. I mean, I know Gabriel somewhere here has done some work on motion, but in terms of actually using motion to recognize, no. Wanted to do it.
So lots more to do for all the grad students out there. Okay, thanks so much, Cathy. So our second speaker is Ferrinne Spector, and she completed her PhD with Daphne in 2009, and she's now an associate professor of psychology and neuroscience at Edgewood College. So she studied development of multisensory perception, so you get another bit of what Daphne spent some of her research doing. And she focuses on learning and natural biases. So she asks interesting questions such as, where does the bias come from to associate visual color lightness with pitch height? And what does that tell us about visual development, learning, and how the brain is organized? So currently, she uses VR and neurophysiological measures to study these questions. And I should also note that she's an award-winning teacher. She was awarded the 2021 Under Koffler Excellence in Teaching Award, as well as the Honored Instructor Award in 2022. And I'll also say she takes very seriously the challenge to validate our tools and our results across populations and cultures. So maybe we'll hear a little bit about that, we'll see. So actually I'm going to sum up Ferrinne in her own words, in which I think you can clearly see Daphne's influence. So she loves nothing more than a well-controlled experimental design and hanging out with dogs. She lives in Madison, Wisconsin with her family, where she spends a lot of time answering questions about brain development over dinner. So, Ferrinne.
Perfect. Okay. All right. Hey, everybody. Hello. Welcome. And thank you, Laurel, for the wonderful introduction. It is so cool to be back here at my alma mater with my former colleagues. Mayu, I'm so happy to see you. Look at these beautiful faces. It was such an honor. Oh, this is exactly the picture I wanted. Oh my goodness. I was actually going to show this picture and say, look at this amazing stack of data that we now have on like Elliott's little computer there. There it all is. So that is very cool. So one thing that I did, I'm sorry, let me be clear. I am not giving a research talk right now, okay? There's a number of reasons for that. One, we got 6 talks. I sort of thought maybe we'd break it up a little bit. But also, for me, the legacy that Daphne Maurer had for me and my career, yes, is about research, but it's bigger than that. It's about mentorship. It's about science writ large. It's about teaching. It's about student support. And it is about training the next generation of, as Cathy says, right, our next generation of scientists, clinicians, anybody who is interested in right? That is the term. That is what my role is, because I learned an awful lot about experimental design across the lifespan. Not only children, also adults, and data collection, all of these things, right? These things that you think when you are a PhD student, this is what you need to learn. You need to learn how to be a scientist. But that's not the only way to be a scientist, is to collect the data. And this is another soundbite in my head. Daphne Wisdoms, right? I'll call them Daphne Wisdoms. She said, look, here is my job as a mentor. You need to do these things, and I will feel as though I have trained you for whatever your job is going to be. I mean, that's almost a direct quote. Was I the only student you ever said that to? Probably not, right? Did she ever say this to you? I don't know. She did, right? First, you need to learn to do science, obviously. Second, you need to give a scientific talk to a scientific audience. Coggy talks, anybody? You still have those?
They're called pizza lunches.
They're called pizza lunches. Well, I absolutely use the term coggy in my classes. I'm like, oh, this is real coggy. Now my students know what that means, by the way. They say, thank you, Bruce. And, okay, you gotta have a scientific talk. 3, you have to give a public talk. You must know how to talk to the public. You must know how to take your science and scale it appropriately so that people can understand and that they are not put off, right? By you just being, right? And she said, I mean, there was publications in there. Publications, okay, 5 things. Publications, obviously, I did those. And then you gotta learn to teach. So you gotta learn to teach. You have to learn to teach. I know people who go through whole entire PhD programs and never teach, never mentor. And I think it is so beautiful. And guess what? These people, they go and they find themselves in their first undergraduate classroom and they're like really intimidated. Because a classroom full of undergraduate students is actually a lot more intimidating than a whole lecture hall full of scientists who are all interested in tearing apart these statistics. And so the first moment is to, I think, that like a lot of mentors, wonderful, excellent mentors in this program, I think the idea is that you are training people, the next generation of you. And I think it's beautiful, it's so nice. It wasn't what I wanted. It just wasn't. And I realized this, and this is another Daphne thing. I realized this when one day, Daphne was like, okay, great. In the past month, there have been 100 papers on it. I don't remember them. There have been 100 papers on our topic. I can't wait to talk about them. And she's like, okay, read them, and then we'll talk about them. And I was like, okay. And I was like, you know what? I'm not looking forward to this. Actually. More on the teaching training. I was doing the science training, but I wanted to emphasize the teaching training. And when I approached Daphne with this, not that she was concerned about me wanting to do teaching training, but maybe what I wanted to maybe do with my training was focus more on undergraduate teaching and mentorship. And I suspect that she had an internal monologue that she did not display. But she responded exactly as one should. Well, I think you're capable of doing anything that you want. But it is in fact your life. And you've got to do the thing that is the best for you. So how can I help you in that path? Which is, it's beautiful. I mean, there are these moments in my life where Daphne is the angel on my shoulder. Occasionally the devil on my shoulder, but mostly the angel. And I think about these moments in life where you make decisions or you don't, and things just happen. It just happened to come. It just happened to work with what I think is the best mentor in the history of Canadian research. Or American research, or worldwide. Every single day. I mean it. And really not exaggerating. I talk about Daphne, not specifically. I talk about her work in class. I draw pictures like this. No, not that. I draw pictures like this. I just drawing a face like St. Elizabeth. That's probably what it looks like here. You'll see it better. I say something like this. I say, what's the difference between this and whatever Cathy just drew? Look at this. You know, I say something like this. And they're like, oh, yeah, well, that one's face-like. And I'm like, yeah, babies are really inclined toward that. I talk about experience-expectant development and experience-dependent development. And if you ask my students, if you said, okay, what are Ferrinne's catchphrases? They will say this. They will say, experience drives brain development. That is what they say. Ferrinne says, experience drives brain development. Where did I get that? Right? How did I know that? How did I come to teaching with this deep and abiding knowledge and truth? It came from you. It came from your body of research. It also came from what I watched you teach in the classes that you were teaching, how you talk to your students. The other thing that they will tell you, that I say a lot, is that categories vary in the brain and behavior. Now that's not something I ever heard Daphne explicitly say, but I think that implicitly we learn how to critically think about behavior, about brain, about development. And when you know how to critically think about things, you can draw conclusions that are really, far-reaching. So the other thing that happens, and this is when I do explicitly describe my mentorship relationship, it is when I have students who are interested in going to graduate school, even when they're only interested in applying for a job. And I say this, I say, make sure it's the right fit. And then I explain some other things. Make sure it's the right fit. And then I describe the experiences that I just described to you. And I say anything less than that is not going to be the right place for you. They are coming from a place where they receive support and mentorship that is flexible and sensitive. They receive support and mentorship that is collaborative, right? It's not just me telling them what to do. It is us working together, right? I didn't just magically know how to do it. I learned how to do that. I learned how to do that by watching. I learned how to do that by engaging. I learned how to do that by seeing what other mentorship structures look like. Not everybody has this best fit, right? And not everybody would fit best in this environment. My students know who Daphne Maurer is. They do. Every single one of them. They know what COGI means. They know what experience-expectant development is. And that's not an accident. I don't intend to do it. I wasn't like, oh, I'm going to talk about Daphne Maurer all the time so that I'll be invited to give a talk at her induction as the officer of the Order of Canada. I don't actually know what the Order of Canada is. I know it really matters. I don't do it intentionally. I do it because that's how I learned. I learned that this idea, this support, this mentorship, this legacy is infused in every aspect of one's life. And I have very important relationships in my life. And although our formal mentoring relationship is 15 years past now, I'm still invoking this every single day. Well, if that's not correct, every day.
Well, thanks for that very inspiring talk. I don't know if that is the kind of talk anybody wants to ask a question about,
It's not a big question.
but actually maybe there is, maybe you've actually opened them to think about things in a different way. And does anyone have a question? Yes.
Hi, thank you so much. That 24 trials episode was really inspiring, and I wanted to ask, in some labs there are seniors who are already in front of you, and if you go out and ask for resources, you can actually get the answer faster. But how do you think we should foster the independence while
Okay, that's an interesting question. I think what I do for my mentorship structure is we meet. I'm not gonna lie, based on Daphne's work here. We meet and we say, okay, what are your big questions? What are you interested in, right? And then they say, oh, I'm generally interested in these really broad things, right? Then we need to rein it in a little bit. And so I say, okay, we'll focus on the ones that you are the most interested in. You're always going to want to do what you're most interested in, right? That's always true. And then I say, okay, go out and read a bunch of literature on those things, right? They don't have to read it in great detail at this level. They just got to come back and have sort of put it all together. What are other people asking? What have they found? What kinds of methods do they use, right? You can't ask ChatGPT that question. Right. Then we come back, right? And then each meeting we start sort of trying, I describe it like are there any bee people in here? I describe it like the bee waggle dance, right? Where it's like you start real big, or like one little tiny bee is like, it's in that direction. And then there's more and more and more bees, and then everybody's like, that's the one, right? And so we realize that we have found the hive when the student is like, boom, I've narrowed in from this really big thing into these really, specific methodology. And then we go forward from there. So I think that, I'm not sure I'm answering your question, but I think that there are so many resources out there. And empirical resources are the only ones you're supposed to be using, of course. You know what I do a lot of, honestly? I do a lot of teaching how to search for the right things. That's actually a very common thing I spend time doing. My students will call me a Google Scholar magician, right? Because they'll spend hours looking for something, and then I'm just like, and then I make sure I tell them, this isn't magic. This is a skill I have learned, right? This is a skill I have learned. Same with understanding how to take big ideas and turn them into experimental studies that you can carry out. It's not magic. It's a skill I've learned through rigorous and intentional training. Again, I don't know that that's answering your question, but when I've got a senior in front of me and they're like, I want to do a project, I'm like, great, we're starting with an absolutely blank slate, right? And so you've got to fill in those blanks. My job here is to make the foundation, right? The scaffold, the structure. You've got to decorate the building and make all the finishing touches, right? Okay, I guess that's cool.
Great, thanks very much. So the final student that was mentored by Daphne is our very own Mayu Nishimura, and she completed a PhD with Daphne in 2008, studying, surprise, the development of face processing. She worked on a number of projects after that, and I just wanted to highlight one that was actually back with Daphne. So they worked on a large project setting up visual screening for young children through the schools, and I remember that Mayu I used to see her with all this equipment. She was traveling all over Ontario to all these schools testing children's vision with the goal of identifying children early who had correctable, visual issues that so they could see the world properly from a very young age. So, it was an incredible, huge study, and you were one of the people that really made it happen. So after that, we hired you. So we hired you as an assistant professor, which you are now here at McMaster. In our teaching stream, you're a very sought-after instructor, already won awards for your teaching. And I'll say at the same time, you've published over 30 research papers, which I think is pretty impressive. I have to say that you are really an amazing colleague. You always have a smile on your face. I love it when I walk down the hall and you're there. Give a little pep talk. Yes, everything's going to be good. And you've very enthusiastically spearheaded so many things in our department that have made it a better place. So thank you, and we look forward to your talk.
Okay.
This one's dead. It is, yeah. There are some like seats here. If any of you want to move down? No? You're good? Okay. So I should have known what Ferrinne was going to do and really placed her last, right? Because like I don't have that kind of entertainment value, but it kind of makes sense because I think my talk lands somewhere in between Cathy and Ferrinne's. But also hearing their talks made me realize that, I thought I was special in how Daphne was mentoring me, but it turns out she does this to everyone. So what have I learned? I've learned so much from Daphne that there wasn't, I don't have a very good cohesive research story like Cathy, and I can't just talk, freely like Ferrinne does, but I did find a few quotes that kind of highlight basically what I learned from Daphne. So that's how I've tried to structure my talk. So the first one is, if at first you don't succeed, try again. All right, so this is the Bobo study. Yes, some of you will recognize it. Oh, some of you are thinking it's this one, the Bandora's Bobo doll study. Yes. This one is almost as famous as the one I'm going to tell you about today. This is the Bobo study, okay? So you might think, what are these? Well, it's helpful that Cathy was here because she already told you about the Jane sisters who differ only in the spacing of features. And my question was, this was around the time I'm coming into Daphne's lab, Cathy's there, they're very excited about all the things that we can learn about face perception. And my question is, spacing takes a really long time to develop. What's special about it? And does it have to be faces? Could it be anything else as well? So I'm saying this is weird. Okay, so I recreated Jane's spacing as just 4 blobs, right? And my question was, can you tell that the spacing of the blobs are changing depending on whether we prime this so that you see the blobs as either facial features or a letter, like the points of the letter Y, right? So I'm quite proud of this research design. And my very first experiment, my first year at Mac, I show after you've been primed to see these 4 blobs as either faces or letters, I take away the priming, and now I ask you, are those 2 pictures the same or different? And lo and behold, the accuracy is much better if you're in the face group than the Y group. Something is special about faces. So then I want to get to the kinds of studies that Cathy's doing. Can 6, 8, 10-year-olds also show that this is much easier to do if you're convinced that these blobs are faces, not letters? What happened next? Okay, study 2, study 3, study 4. These are the 3 words that PhD students fear the most. Failure to replicate. Okay? I don't know, I'm doing all kinds of different manipulations. I'm trying to perfect the paradigm so it's ready to be tested with kids, and yet I cannot recreate this effect. So here's Daphne's mentoring moment. Do you still want to publish the results from experiment 1? And this is kind of what Ferrinne and Cathy were saying. Daphne is recognizing and acknowledging that this is my study. I had the idea, I was excited about it, and if I'm going to pull the plug, it's going to be my decision, right? So I'm thinking about it. She's like, we did get that effect the first time. I'm not denying that. But I said, yeah, but how are we going to explain how we never got it again, right? So I'm thinking, maybe, how about a different question? And I'm pretty sure Daphne was relieved that that was my choice, right? Because right there, Daphne could have mentored me to just publish. Right, we need more publications. Or she could have told me, we cannot publish this, this would be irresponsible. But instead, she let me choose. So I said, maybe we ask a slightly different question. So as Cathy alluded to, when we actually learn faces, we're learning individual faces, right? We don't really want to confuse Sigal and Pat. They are 2 different people. although you might confuse them some days. So what if I name these, now I had to make them upside down so they're not really face-like. And I'm going to teach you that this is Bobo 1, Bobo 2, Bobo 3, 3 different Bobos. I need you to learn individual Bobos. Now I can create 2 different groups. People who learn Bobos just at the basic level of categorization, so know the difference between Bobo and Gigi, Tika, Lula, all of these things are just blob-like shapes. But there's a group that has to learn the individual Bobos. And when we do that, at the pre-test, the individual group and the basic level training group are performing similarly. But after the training, it turns out if you have been taught to differentiate the Bobos at the individual level, a new set of Bobos, you can actually tell them apart. Now, unfortunately, 24 hours later, the effect is gone. So it's really transient. And we even did a second study to make sure that we can replicate those findings, right? Even with a control group added in. So that was my first publication with Daphne. And so now I know, and this is kind of a common theme from Daphne's lab, that experience determines perceptual sensitivities. If that spacing difference is important to learn, you learn it. If it's not important, you don't even notice, right? And so I'd like to modify this first quote, in fact, to be, if you fail, switch to a new topic. Okay? You don't have to keep at it. All right, so the next portion is, I think, exemplified by this quote. Greater even than the greatest discovery is to keep open the way to future discovery. And one of the things that I love about Daphne is that, there are some brilliant researchers who want to own everything, right? This is my lab, my data, my paradigm, my students, right? And they're not willing to share their curiosity, their knowledge, their resources. Daphne is the opposite. If she sees a good idea, she runs with it. She thinks outside the box, as Cathy said. So Daphne goes to do a sabbatical in Australia, comes back and says, I think you'll learn a lot by working with Gillian Rhodes and Linda Jeffrey, because they are asking very interesting questions that I think align with your questions, a good fit in terms of your research interests. So I want you to stare at this face. Solid 5 seconds. Keep staring at it. Keep staring at it. Here's a target face. You only get to see it for a split second. Now, did it look more like Jim or more like Dan? Hands up for Jim. Hands up for Dan. Okay, you are all horrible participants. It was supposed to look more like Dan. So now I'm not sure what to conclude from this. You weren't paying attention, or one trial is not enough. Okay, so once you do this for 24 trials, right, and you're not 2, so you can handle 24 trials. So this was called the face identity after effect. So okay, you didn't experience what I had hoped you would, but now that we're in the age of digital photographs, you can create landmarks on the faces, and you can recreate the average face from a whole bunch of faces like Dan. But what you can also do then is, in every way that Dan deviates from the average, like having a big forehead or a strong jawline, you can recreate the difference in the opposite direction to create an anti-Dan. Right? So anti-Dan has a smaller forehead and a more angular jaw. In every way that Dan differs from average, anti-Dan is the opposite. And the idea here is that maybe the way we encode these individual faces is by somehow processing these deviations from average, your average of all the faces that you've seen. If this were true, when you adapt to anti-Dan, it pulls the average face more like Dan, anti-Dan, because you're being exposed to it, so you're more likely to call this average face Dan. But the interesting thing is that if you adapt to anti-Dan, it should pull your perception towards Dan. That's a match trial. But if you adapt to anti-Jim, Jim is on some other dimension, right? Jim is not Dan. So if you adapt to anti-Jim, you're not more likely to call this Dan. In fact, you should be more likely to call the face Jim, right? So there are match trials and mismatch trials. And what we found is that with adults, so here's mean proportion correct, so that's being able to say that the face is Dan, even if it's only 30% Dan or 60% Dan, and that's the middle dotted line. In the match trials, your perception is pulled towards Dan, so the line's above the dotted line. And in the mismatch trials, your perception is pulled in some other direction, so you're less likely to call that face Dan. So that difference is the identity after effect. And it turns out that 8-year-olds pretty much show the same pattern, right? So you heard Cathy say, some of the things about face perception develops really slowly. It can be shown at 6-year-olds, 8-year-olds, 10-year-olds. Well, 8-year-olds already show a face identity after effect, leading us to conclude that children already demonstrate these norm-based processes. Okay, and you get to present these findings at conferences. So there's me and Ferrinne of 20 years ago, right? We look exactly the same. And some other lab mates, there's Katie from Laurel's lab, Paul from Lewis's lab. So this was the developmental group at the time going to conferences together. And so next, here's another example of Daphne kind of elevating her students. So Marlene Berman from Carnegie Mellon comes to give a colloquium talk, and she does really cool research with prospagnostics. So these are patients who cannot recognize faces. You show them a picture of a face, they know that they're looking at a face, but they can't figure out who it is. So I loved her colloquium talk, and Daphne afterwards says, Marlene, are you looking for a postdoc? Because Mayu here is about to finish, and she's amazing. And Marlene says, why, yes, I am looking for a postdoc. So here I go off to Pittsburgh, and this is late 2000s. So there's a big hype about fMRI research. Right? It's at a time where it's almost like if you are serious about doing psychological research, you have to know how to use fMRI. So off I go to Carnegie Mellon to start asking questions related to face perception, but I was not convinced at this point now that faces are in fact really special as faces. So I was like, I just want to know more about object recognition in general and how it develops. So a good model for looking at object recognition development is the area of the brain called the lateral occipital cortex, LOC. It responds preferentially to objects than any other kind of visual stimuli. But you'll notice here in this graph that there's the LOC in adults, adolescents, and so it does exist in children, but it's much smaller. So my question is there some development in the LOC and how it responds? So viewpoint is important in recognizing faces. So if you know the person, you will be able to identify that person regardless of changes in lighting, makeup, viewpoint. Daphne doesn't know who this is. Oh, she does know. Yes. Right, because she was just in Toronto, right? So how does this view invariant, the fact that you can recognize the same person from different views develop? That was what I was curious about. But I want to compare children versus adults, right? Fundamentally, I'm interested in developmental questions. So any object you use, it's confounded with age and experience. Adults will have seen those objects more than children, so I had to create new objects. The funny thing about looking at objects is when you're presented with the same object, it's really not exciting. If I change the size, okay, for a moment you might be like, oh, well, at least it's a little bit different. But if I keep showing you the exact same shape, all in different sizes, it doesn't take very long for you to be like, this is very boring. You're showing me the exact same thing, right? So this is size invariance. We're very good at knowing that it's the same object even when the size changes. And if you see the same object, but now you see a different object, it's a bit more exciting, right? Your brain recognizes that, oh, this is new information, maybe we should pay attention. So Girl Spector and Nancy Canwisher had just recently published a study where they showed that if you show the same picture, whether it's identical pictures or the identical object in a different format, that is a photograph versus a line drawing, you actually get neural adaptation. That is the bold signal, right? The oxygenated blood is going to be less if you repeat the same object. You don't get that adaptation if you show different objects. So you can ask participants, do you consider these objects to be the same or different? Or you can also look at the neural activity and see whether you can see differences in the brain signals. So that's what I wanted to do. Okay, so I had to create new shapes, right? And so at the top is the exact same shape from the same view. So I expect maximal neural adaptation there. Boring, boring, boring. The bottom row is all different shapes, minimal adaptation, right? We should see high activation for those. Now, I'm just creating these shapes on my own, like Ferrinne said, on the computer, just creating 3D shapes. It turns out that I had unintentionally created some shapes that were view dependent. So the second row there, pilot testing revealed that participants said those shapes look different to me depending on the view. Whereas this third row are more view independent. That is, participants were more confident that these were the same shapes even when you change the view. So I did end up creating some objects that were more view independent, some objects that were more view dependent. Bottom line, though, I'm interested in developmental questions. So do we see neural adaptation across age? So our youngest participants were 5-year-olds. On the y-axis is the adaptation index, and the dots, the circles, are individual participants, and you can see most of the dots are above 0, so we do see adaptation, but there's no effect of age. The line's flat from age 5 to about 25. Whereas with view variance, right, when you change the viewpoint, we start to see this correlation. So that the youngest kids, we actually don't see neural adaptation when it's the same object, but you change the view. It's almost as if the brain signals are treating it as a new object, right? Whereas when you're older, you do start to adapt even when the view changes. And this nicely supports Cathy's claim, right, that kids also have difficulty identifying faces when they change different shapes. But here's a bit of a identity crisis for me, because what do we do with like this 25-year-old who doesn't show any view adaptation, right? And some of the 5-year-olds are showing adaptation. So this is the tricky thing about fMRI, is that it's such a blunt tool, right? So kudos to all of you who do fMRI research, but here's the time where, kind of like Ferrinne was saying in her career, I'm thinking, what is this signal, right? I'm not sure if I want to use this tool. But I mean, one conclusion was that certainly there are neural correlates of object recognition that develops through childhood and adolescence. This is something that is changing in the brain through adolescence. Now, one cool study that we did that I don't know exactly what to make of it, so I'll just throw it out there for you guys, is the case of a prosopagnosic patient, and patient SM. He's actually agnosic, so he also can't recognize objects. So interestingly, like if you show a picture of a cow, he might call it a horse. So he can kind of sense that it's an animal with 4 legs, but he really can't tell them apart. Here's patient SM, here's an age-matched control participant. If we look at brain activation while they're looking at objects versus nothing, a blank screen, you can see that the activation patterns look fairly similar in the two brains. But if we do objects versus the scrambled version, you know, all of you can see that there's a clear object here, no object on the right. There's lots of brain parts that respond preferentially to the object, and notice in patient SM, the activation is much smaller. The areas that respond to the object is much smaller. what was cool was, okay, so I talked to you about lateral occipital area. It seems like early visual pathways, early parts of the visual cortex, like V1, V2, V3, are intact in patient SM. So we're going to overlay this grid just on LOC, the part of the brain that's supposed to prefer objects, and compare activation in this patient versus control participants. So the first test was, again, objects versus blank. And here is the entire control group, about 8 adults, a particular age-matched control participant, and SM in the middle. And this is a heat map, so yellow means much bigger difference. So SM, although you see activation, it's weaker than the control participants. That black square in the middle of the right hemisphere is where SM has his neural injury. He was in an automobile accident at age 18 that damaged that part of his brain. What happens when you do object versus scrambled objects? Notice how in SM there's much less activation compared to the age-matched participant and the whole control group. But here's the peculiar result. Let's look for adaptation effects, right? So you show the same object, you should have reduction in the neural signal. All over LOC in the control participant and the control group, you see those adaptation effects. You don't see it in SM. And note, you don't see it in the structurally intact left hemisphere. Nothing damaged his left hemisphere, yet even in the left hemisphere, we don't see adaptation effects, right? So neural injury can impact the contralesional brain region in an adult. All kinds of studies to suggest that the other side can sometimes compensate for damage to one side of the brain, but I think most of those studies are with younger patients, right, when they're children or maybe adolescents. And maybe as an adult, I don't know why. I'm still puzzled by this. So there's something we don't quite understand about the homologous brain regions. You can injure one side and ruin the activation in the other side. I think that's pretty cool. But, and by going to Carnegie Mellon has a Qatar campus, so I also got to teach in Qatar for half a semester. And so there's my mom visiting me in Qatar, so you can ask me about that experience at some other time. Okay, and then the last external mentor I want to talk about is Noah Ofen. So again, as Daphne, tends to say to random people, I think we're a good fit, as she did to Ferrinne. Me being mentored by Daphne, I was living in Michigan, so Noah was at Wayne State University at the time. Now she's at University of Texas at Dallas. I said, Noah, I think our research interests are a great fit. You should hire me as a part-time postdoc. And I realized Cathy started in Daphne's lab in much the same way. And Noah said, excellent, you should come join my lab. So I'm a little bit suspicious about fMRI data at this point, right? But thankfully, Noah does structural research. So at the time, all she was doing in her lab was measuring hippocampal volume. How big is your hippocampus? So that's an example of your hippocampus. We're going to check the size at those slices and extrapolate to give each individual a hippocampal volume. And some cool studies that we found, so hippocampal volume correlates with your socioeconomic status, but only among children, not adults, right? So maybe early in development, nutritional factors, stress factors are much more impactful in brain development and brain maturation than if you're an adult. Maybe by the time you're an adult, there are some compensatory mechanisms to make up for that initial deficit. This is just a correlational study. I don't know exactly why, but still pretty interesting. And the fact that when controlling for age and sex, hippocampal volume does correlate with better memory development. So having a bigger hippocampus does seem to correlate with your memory performance. So things I've learned about object recognition, again, early experience may be more impactful than later experience, also mentioned by Cathy and Ferrinne as well. All right, so the final quote that I want to end with is, alone we can do so little, together we can do so much. So it's just happened that the stars aligned, that I find myself back in Toronto with Daphne on our next project. You can see how Daphne hasn't really understood how to take selfies, right? So she's not really going to do well as an Instagram influencer as her next career. But it was Daphne as a basic vision scientist and Agnes Wong as a clinician, ophthalmologist at, in-chief at Sick Kids at the time, came together to say we should do something about children's vision because early experience matters and early treatment is more effective. So at the time we look at data like this, only 6% of Ontario children having an eye exam. This is bad. So let's, as Ferrinne said, critically evaluate the efficacy of a school-based vision screening program. So we went, as Laurel mentioned, all over Ontario. Is Sally here? Oh, she couldn't be here, okay. Daphne was at a lot of these cities as well, but Sally and I traveled the farthest, so this is at Laxo First Nations. There were lots of helpers at all kinds of schools. There's Daphne also doing the screenings, you know, helping kids get their glasses. I was gonna say, we even had Firbit the mascot, and that's Sally inside. Right, so she's doing her part advocating for wearing your glasses, and you can kind of see me in the corner there carrying boxes, so I was also helping. And we were very careful with our experimental design, so we got the data published in prestigious medical journals like British Medical Journal, Canadian Medical Association Journal, but I think the most impactful thing that happened, now don't share this beyond this room, but Daphne took one version of our report as a printout and accosted the Minister of Health at the time at a public health conference and said, this is really important, you have to read this. And I'm pretty sure that's how we got to this point, where the ministry did update their public health standards so that we now actually have mandated kindergarten vision screening in Ontario, right? Yeah, so alone we can do so little, but together, so Daphne, me, Sally, and Agnes, these were 4 women who were dedicated and with strong conviction that we have to do something, and you can make it happen, right? So I do like this picture, just like Ferrinne loves it, because there's Daphne wrapping up her PhD thesis with all of those boxes full of data. But I think the curiosity that she had then about, you know, all of these interesting questions and what can I do with them, that hasn't gone away. She still carries that, and I'm doing my part to kind of, again, mentor the younger students about their curiosity. So just as one example, one of my students, Amal and Sophia and Jane, worked on this, presented their research at the Innovations in Education Conference happening today. So she presented that today. So it's kind of nice to be given the opportunity to kind of reflect on where I've gone and all the amazing things that Daphne has taught me. So thank you, Daphne, and thank you for your attention.
Well, thanks so much. I think we're out of time, but this was really an amazing journey of some of the contributions Daphne's made in mentoring, and thanks to all our speakers. So we'll have a break now, short 15-minute break, and we'll come back. So if you have questions for Mayo, Mayo should be out in the lobby. Thanks.